Hi, I'm Junique Maynard, and today we'll be discussing, through my flowchart, the synthesis of testosterone, insulin, and cytoskeletal protein. First off, we'll start with the synthesis of testosterone. Testosterone synthesis begins when the luteinizing hormone binds to a Leydig cell transmembrane receptor, which initiates ATP conversion to CAMP, and therefore CAMP initiates the production of protein kinase A. So the first enzymatic reactions takes place when protein kinase A transport cholesterol, which is a substrate for testosterone, in transport vesicles to the inner membrane of the mitochondria. So cholesterol is then converted to pre-pregnenolone by a P450 enzyme, where the steroid synthesis center is in the small endoplasmic reticulum. So if you follow along, pregnenolone is then excreted in secretory vesicles, and the remaining enzymatic reactions takes place. So testosterone is able to cross the plasma membrane bilayer through transport out of the Leydig cells into small vessels of the testes through passive transport, crossing the cell membrane. So, to note, testosterone has a very short half-life, and it is metabolized in the liver, where it is made inactive and expelled from the body through feces and urine. If you follow along with me, we'll go through the synthesis of insulin, which begins in the pancreatic beta cells with the translation of insulin mRNA to its single-chain precursor, proinsulin. The proinsulin is generated with a single peptide through insertion into the endoplasmic reticulum, where it is folded to form 3 disulfide bonds. It is then secreted in transport vesicles. The folded proinsulin is secreted into the Golgi apparatus, where further synthesis occurs. So as blood glucose levels increases, we have insulin being secreted from the Golgi apparatus in secretory vesicles in granules, and it is then transported out of the cell membrane into the bloodstream with the increase of blood glucose. However, after insulin has served its purpose in the body, the liver is known to be the main site of the degradation of insulin by endocytosis, which is a form of degradation of insulin by a degrading enzyme. Lastly, if you follow me through the synthesis of cytoskeletal proteins, which is a rather simple process, we will begin with the transcription of DNA in the nucleus. And then we have strands of DNA that are connected to mRNA, which exits the nucleus to its pores into the cytoplasm, where these mRNA attaches to the free ribosomes in the cytoplasm. So the mRNA directs the needed tRNA proteins for synthesis. The protein synthesis takes place in the free ribosomes. However, unlike the 2 other syntheses we would have mentioned before in our flowchart, such as the synthesis of testosterone and the synthesis of insulin, cytoskeletal proteins, however, does not go to further synthesis through the endoplasmic reticulum nor the Golgi apparatus. Final synthesis of cytoskeletal proteins takes place in the free ribosomes, and it does not exit the cell through secretory vesicles and leaves the cell membrane. Thank you so much for going through with me in my flowchart presentation.